Translational proofreading. Mechanisms and importance in protein synthesis. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Translational proofreading is a crucial process that ensures the fidelity of protein synthesis by minimizing errors in the translation of the genetic code into proteins. During translation, ribosomes read the mRNA sequence and synthesize proteins by incorporating the correct amino acids into the growing polypeptide chain. However, the translation process is not flawless, and errors can occur, leading to the incorporation of incorrect amino acids, which can result in malfunctioning proteins and potentially disastrous cellular consequences. Translational proofreading involves multiple layers of error-checking mechanisms to maintain accuracy, such as amino acyl ternus selection, peptide bond formation, and termination. 1. Role of amino acyl ternus synthetase in proofreading. The first layer of proofreading in translation occurs during amino acylation by amino acyl ternus synthetases. These enzymes attach the correct amino acid to its corresponding terna, a process that is critical for accurate translation. To ensure the accuracy of this process, amino acyl ternus synthetases possess editing sites that can remove incorrectly attached amino acids. This proof point is essential because even a single incorrect amino acylation can result in the incorporation of the wrong amino acid during translation. Initial charging. In the first step, the synthetase attaches the amino acid to the terna. Editing mechanism. If the wrong amino acid is charged to the terna, the enzyme can hydrolyze the mischarged terna, preventing its entry into the ribosome. This editing step ensures that only the correctly charged terna enters the ribosome for translation. This proofreading by amino acyl ternus synthetases is highly efficient and occurs before the terna enters the ribosome, making it the first line of defense against errors in translation. 2. Proofreading mechanisms at the ribosome. Once the correctly charged terna enters the ribosome, further proofreading mechanisms are involved in ensuring that the correct amino acid is incorporated into the polypeptide chain. These mechanisms are largely dependent on the structure and function of the ribosome. Codon-anticodon interaction. The ribosome reads the mRNA sequence in triplets codons, each of which corresponds to an amino acid. The anticodon of the terna pairs with the codon of the mRNA. This interaction is critical for ensuring that the correct terna is selected for the correct codon. Selective elongation. Once the terna is in place, the ribosome facilitates peptide bond formation. If the terna's anticodon does not match the mRNA codon with high fidelity, the elongation process is delayed, and the incorrect terna is rejected from the ribosome. This process acts as a second proofreading step, ensuring that only the terna carrying the correct amino acid is incorporated into the grown protein. 3. Mechanisms of proofreading and elongation factors. In addition to amino acyl ternus synthetase in the ribosome, the elongation factors such as EF2 in prokaryotes and EF1A in eukaryotes play a crucial role in translational proofreading. GTP hydrolysis. Elongation factors help deliver the charged terna to the ribosome. The elongation factor bound to GTP first ensures that the terna is correctly matched to the mRNA codon. Once the terna is properly positioned, the elongation factor undergoes GTP hydrolysis, which changes its shape and releases the terna into the ribosome. If the terna is not correctly matched with the codon, the elongation factor will prevent its release and block the peptide bond formation. Proofreading by elongation factors. This GTP hydrolysis-based proofreading ensures that only the correct terna is incorporated into the growing protein chain. 4. Peptide bond formation and proofreading. Peptide bond formation is another critical step where proofreading occurs. After the terna is positioned correctly in the ribosome, the peptidyl-transferase center catalyzes the formation of a peptide bond between the grown polypeptide chain and the new amino acid. If an incorrect terna has been incorporated, the peptide bond formation is hindered. This proofreading mechanism ensures that the amino acid sequence remains accurate during protein synthesis. 5. Termination and proofreading. The termination phase also involves proofreading to ensure that the protein synthesis process stops at the correct point. Release factors such as RF1, RF2, and RF3 in prokaryotes and ERF in eukaryotes are responsible for recognizing the stop codon in the mRNA and catalyzing the release of the finished polypeptide chain. If the translation machinery encounters a nonsense mutation, a premature stop codon, the proofreading mechanisms involved in ribosome dissociation and release factor activation ensure that translation terminates at the correct location. 6. Proofreading and quality control mechanisms. Besides the direct proofreading steps in the translation process, there are quality control mechanisms in place to handle errors in translation. One such mechanism is the translational fidelity checkpoint, which involves the surveillance systems like non-stop decay (NSD) and no-go decay (NG) pathways. These systems detect and resolve errors such as stalled ribosomes or frame shift errors during translation. Previous year questions. PYQs 1. CSIR Net Life Sciences 2017. Which of the following factors contributes directly to ensuring the correct codon-anticodon matching during translation? Of amino acyl ternus synthetase. B. Elongation factor G. EFG. C. Ribosome-associated release factor D. Peptidyl-transferase activity. Answer. Of amino acyl ternus synthetase. Explanation. Amino acyl ternus synthetase ensures the correct amino acid is attached to the terna, which directly affects the codon-anticodon matching during translation. 2. CSIR Net Life Sciences 2019. What is the primary role of elongation factors in translational proofreading? A. To charge ternas. B. To ensure the correct codon-anticodon matching. C. To catalyze peptide bond formation. D. To release the newly synthesized polypeptide. Answer. B. To ensure the correct codon-anticodon matching. Explanation. Elongation factors like EF2 in prokaryotes ensure the correct terna is selected for the codon in mRNA, promoting translational accuracy. 3. CSIR Net Life Sciences 2020. Which of the following best describes the proofreading mechanism that occurs during amino acylation of terna? A. Editing by the ribosome. B. Editing by release factor. C. Editing by amino acyl ternus synthetase. D. Editing by elongation factors. Answer. C. Editing by amino acyl ternus synthetase. Explanation. Amino acyl ternus synthetase has an editing function that removes incorrectly attached amino acids from terna before they enter the ribosome, ensuring the fidelity of translation. Conclusion. Translational proofreading is a crucial aspect of protein synthesis that ensures the accuracy of protein production. From amino acyl ternus synthetase editing to ribosomal codon-anticodon interactions, and the role of elongation factors, each step is designed to minimize errors and ensure that the genetic code is faithfully translated into functional proteins. The effectiveness of these proofreading mechanisms is essential for the proper functioning of the cell and is an important concept for mastering the CSIR Net JRF Life Sciences exam. Understanding these proofreading systems will not only help in answering theoretical questions but also in application-based questions during the exam. You said. Translational inhibitors. ChatGPT said. Translational inhibitors, mechanisms, types, and relevance in cellular regulation and medicine. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Translational inhibitors are substances that interfere with the process of protein synthesis at various stages. Translation is a complex and energy-demanding process that synthesizes proteins based on the information encoded in mRNA. Translational inhibitors can target different components of the translation machinery, including ribosomes, terna, translation initiation factors, and elongation factors, among others. These inhibitors are important tools in cellular regulation, antibiotics, and cancer therapies, as they can selectively block protein production in specific cells or organisms. 1. Mechanisms of translational inhibition. Translational inhibitors can work at several stages of translation. Initiation. Inhibitors that block initiation interfere with the assembly of the translation initiation complex. This includes the binding of ribosomes to mRNA and the initiation factors, e.g., ACE in eukaryotes. If initiation is blocked, translation cannot begin. Elongation. Inhibitors targeting elongation affect the process of adding amino acids to the grown chain. These inhibitors typically interfere with the function of elongation factors or ribosomal subunits responsible for catalyzing peptide bond formation. Termination. Some transdermal inhibitors work by preventing the release of the newly synthesized protein from the ribosome once translation is complete. This can occur by inhibiting release factors or interfering with the recognition of the stop codon. 2. Types of translational inhibitors. There are various types of translational inhibitors, including those that target prokaryotic and eukaryotic translation systems. Below are the major classes of translational inhibitors. A. Antibiotic inhibitors, prokaryotic translation inhibition. Many antibiotics target the ribosomes of bacteria, disrupting protein synthesis and leading to bacterial death. These antibiotics are particularly effective because they selectively target bacterial translation machinery, which differs from that of eukaryotes. Some common ribosome-targeting antibiotics include tetracyclines. These bind to the 30S ribosomal subunit and prevent the attachment of amino acyl ternate to the ribosome, thereby inhibiting elongation. Chloramphenicol. This antibiotic inhibits the peptidyl-transferase activity of the 50S ribosomal subunit, preventing the formation of peptide bonds during elongation. Erythromycin, a member of the macrolide class, it binds to the 50S ribosomal subunit and prevents the translocation step during elongation, halting protein synthesis. B. Eukaryotic translation inhibitors. In eukaryotes, translation inhibitors are often used in cancer therapies and research to control protein synthesis. These inhibitors may work by interfering with various translation initiation or elongation steps. Cyclohexamide. This inhibitor acts on the eukaryotic ribosome and prevents the elongation process. Cyclohexamide. This inhibitor acts on the eukaryotic ribosome and prevents the elongation process by inhibiting the translocation step, thereby halting protein synthesis. Rocagamides. These compounds interfere with the cap-binding complex A4F in eukaryotic translation initiation, blocking the assembly of the ribosome mRNA complex and initiation of translation. Rapamycin. This compound inhibits the mTOR pathway, a key regulator of translation initiation in eukaryotes. By inhibiting mTOR, rapamycin reduces the translation of proteins involved in cell growth and proliferation, making it useful in cancer therapies and immune system regulation. C. Natural translation inhibitors. Several natural compounds have been found to inhibit translation in both prokaryotic and eukaryotic systems. Puromycin. This naturally occurring antibiotic mimics the structure of amino acyl ternate and enters the ribosome at the A site during elongation. Upon incorporation into the grown polypeptide, puromycin causes premature chain termination, leading to incomplete protein synthesis. Ricins. Ricins are potent toxins from the sugar bean plant. They inhibit protein synthesis in eukaryotic cells by modifying the ribosomal RNA, rRNA, of the 60S ribosomal subunit, causing its inactivation. Ricin catalyzes the ADP ribosylation of a specific adenine in the rRNA, halting translation. Diphtheria toxin. Produced by the bacterium Corynebacterium diphtheriae, this toxin inhibits translation by ADP ribosylating E2, an elongation factor necessary for translocation. This modification prevents translocation from occurring and stops elongation. 3. Translational inhibitors in medical applications. The use of translational inhibitors has significant therapeutic potential, particularly in the development of antibiotics, cancer drugs, and immune modulators. By inhibiting the translation of specific proteins, these inhibitors can halt the progression of diseases at the cellular level. Cancer therapeutics. Translational inhibitors such as rapamycin and cyclohexamide are used to inhibit the synthesis of proteins required for tumor growth and cell proliferation. By blocking these processes, these drugs can slow or stop the growth of cancer cells. Antibacterial drugs. Antibiotics like tetracycline and chloramphenicol are widely used to treat bacterial infections by inhibiting bacterial translation. These drugs selectively target bacterial ribosomes, making them less toxic to human cells. Immunosuppressive drugs. Drugs like rapamycin are used to suppress the immune system in organ transplantation, as they inhibit translation of proteins that promote immune cell activation. 4. Mechanisms of resistance. Mechanisms of resistance to translational inhibitors. Bacteria and cancer cells can develop resistance to translational inhibitors through several mechanisms. Mutations in ribosomal proteins. Bacteria can evolve mutations in ribosomal proteins or ribosomal RNA to reduce the binding efficiency of antibiotics like tetracyclines or erythromycin, rendering these drugs ineffective. Efflux pumps. Some bacteria can employ efflux pumps to actively pump out antibiotics from the cell before they can reach their target, decreasing the drug's efficacy. Modification of elongation factors. In the case of inhibitors like diphtheria toxin or ricin, bacteria or cells can evolve modifications in the elongation factors they use to resist the effects of these toxins. Previous year questions. PYQs 1. CSIR net. Previous year questions. PYQs 1. CSIR Net Life Sciences 2018. Which of the following inhibitors specifically targets prokaryotic translation? A. Cyclohexamide. B. Tetracycline. C. Ricin. D. Puromycin. Answer. B. Tetracycline. Explanation. Tetracycline inhibits prokaryotic translation by binding to the 30S ribosomal subunit and preventing the attachment of amino acyl ternate to the ribosome. 2. CSIR Net Life Sciences 2020. Which of the following drugs inhibits eukaryotic translation by interfering with the A4F complex during initiation? A. Cyclohexamide. B. Rapamycin. C. Rocaglamide. D. Chloramphenicol. Answer. C. Rocaglamide. Explanation. Rocaglamide inhibits A4F during translation initiation, blocking the assembly of the ribosome mRNA complex and preventing translation. 3. CSIR Net Life Sciences 2019. Which of the following about ricin? A. It inhibits translation by preventing peptide bond formation. B. It ADP ribosylates E2, causing translation termination. C. It inactivates the ribosome by modifying rRNA. D. It blocks ternate charging during translation. Answer. C. It inactivates the ribosome by modifying rRNA. Explanation. Ricin inhibits eukaryotic translation by modifying ribosomal RNA in the 60S ribosomal subunit, preventing its function in protein synthesis. Conclusion. Translational inhibitors play vital roles in regulating protein synthesis and have important applications in medicine, particularly in antibiotic development, cancer therapy, and immune suppression. They work by targeting different stages of the translation process, from initiation to elongation and termination. Understanding the mechanisms of action and applications of these inhibitors is essential for CSIR-NET life sciences candidates. Mastery of this topic will help in answering both direct and application-based questions in the exam, as well as in understanding broader biomedical concepts. You said. Post-translational modification of proteins. ChatGPT said. Post-translational modification of proteins, mechanisms, types, and biological significance. CSIR-NET JRF Life Sciences. Maximum detail. Paragraph format. Note tables. PYQs included with year and answers. Post-translational modification (PTM) refers to the chemical changes that proteins undergo after their synthesis during translation. These modifications are crucial for the proper functioning, localization, stability, and interactions of proteins within the cell. PTMs are involved in a wide range of biological processes such as cell signaling, protein degradation, protein-protein interactions, and metabolism regulation. PTMs can occur in response to environmental changes, signals from the extracellular matrix, and cellular stresses, thereby adding layers of regulation to cellular processes. 1. Mechanisms of post-translational modification. Post-translational modifications generally involve the covalent attachment of a functional group to specific amino acid residues of a protein. These modifications are carried out by a variety of enzymes and cofactors that add or remove cis chemical groups. Some modifications occur on specific residues, while others are more widespread across the protein structure. The resulting changes can alter protein activity, structure, interaction with other molecules, and half-life in the cell. 2. Types of post-translational modifications. PTMs are classified into different categories based on the chemical group added or modified. A. Phosphorylation. Phosphorylation is the addition of a phosphate group post-square to specific serine, threonine, or tyrosine residues in a protein, catalyzed by kinases. This modification is reversible, with the removal of phosphate groups carried out by phosphatases. Phosphorylation plays a critical role in cell signaling, regulating processes such as cell cycle progression, metabolism, apoptosis, and gene expression. It is a key mechanism in the activation and inactivation of proteins involved in signaling pathways, including the MAP kinase pathway, PI3K-AKT pathway, and G-protein-coupled receptors GPCR signaling. Example: phosphorylation of histone proteins can alter chromatin structure, influencing gene expression. B. Ubiquitination. Ubiquitination is the attachment of ubiquitin, a small protein, to the lysine residue of a target protein. This process is catalyzed by a cascade of enzymes: E1 activating enzyme, E2 conjugating enzyme, and E3 ligand enzyme. Ubiquitination can serve various functions, with the most well-known being the tagging of proteins for degradation via the proteasome. It is also involved in processes such as DNA repair, cell cycle regulation, and endocytosis. Example: the ubiquitination of cyclins leads to their degradation, regulating the cell cycle. C. Acetylation. Acetylation involves the addition of an acetyl group coached to the amino terminal lysine residues of a protein, primarily catalyzed by acetyltransferases. Acetylation of histone proteins is a key modification that regulates chromatin structure and gene expression. It either promotes gene expression by relaxing chromatin or repress it by compacting the chromatin. Additionally, acetylation is involved in the regulation of metabolic enzymes and signal transduction pathways. Example: acetylation of histones by histone acetyltransferases (HATs) can lead to the activation of transcription. D. Methylation. Methylation is the addition of a methyl group CH to the nitrogen atom of lysine or arginine residues. This modification can regulate protein function by altering its structure, interaction with other proteins, or interaction with DNA. In the case of histone methylation, it can either activate or repress transcription depending on the specific residues modified in the type of methylation mono, D, or trinkylation. Example: histone H3 lysine 9 trinkylation H3K9Me3 is associated with heterochromatin formation and gene silencing. E. Glycosylation. Glycosylation refers to the attachment of sugar moieties, oligosaccharides, to a protein. It can occur on asparagine N-linked glycosylation or serine threonine residues O-linked glycosylation. Glycosylation is crucial for the stability, folding, and function of proteins, especially in the extracellular matrix and membrane proteins. It also plays a critical role in cell signaling and immune responses. Example: glycoproteins involved in immune recognition, such as antibodies, are often glycosylated to aid in their function. F. Sumolation. Small ubiquitin-like modifier sumo proteins are covalently attached to lysine residues of target proteins in a process called sumolation. This modification is similar to ubiquitination but typically regulates protein localization, activity, or stability rather than targeting proteins for degradation. Sumolation plays an important role in the regulation of nuclear cytoplasmic transport, gene expression, and DNA repair. Example: p53, a tumor suppressor, is regulated by sumolation, influencing its stability and function in cell cycle arrest and apoptosis. G. Lipidation. Lipidation involves the addition of lipid groups to proteins, which facilitates their association with cell membranes. It is essential for the localization of proteins to various cellular compartments and their interaction with lipid rafts or membranes. Common types of lipidation include palmitoylation, myristoylation, and prenylation. Example: raw beans are prenylated, which helps anchor them to the inner side of the plasma membrane, a crucial step for their function in cell signaling. 3. Biological significance of post-translational modifications. Regulation of protein activity. PTMs can activate or deactivate enzymes and signaling proteins. For instance, the phosphorylation of enzymes involved in signal transduction can lead to the activation cascade of responses that regulate cellular processes. Protein localization. PTMs like acetylation and lipidation help proteins localize to specific cellular compartments, such as the nucleus, mitochondria, or the plasma membrane. Protein-protein interactions. PTMs can modulate the ability of proteins to interact with other proteins or molecules, thereby influencing cellular processes like transcription regulation, signal transduction, and protein assembly. Cellular stress response. Many PTMs are involved in the response to cellular stress, including heat shock, oxidative stress, and hypoxia. These modifications help in the stabilization or degradation of damaged proteins. Clinical relevance. Post-translational modifications are involved in several diseases, including cancer, neurodegenerative disorders, and inflammatory diseases. For example, mutations that disrupt normal ubiquitination processes can lead to cancer by preventing the degradation of oncoproteins. Likewise, incorrect methylation patterns are often observed in various cancer cells and autoimmune diseases. Cancer. Abnormal PTMs can lead to the accumulation of oncogenic proteins, contributing to tumorigenesis and metastasis. Targeting PTMs involved in cell cycle regulation or apoptosis has become a therapeutic approach. Neurodegenerative diseases. In diseases like Alzheimer's disease, Parkinson's disease, and Huntington's disease, improper phosphorylation or ubiquitination can lead to the accumulation of misfolded proteins, which aggregate and damage neurons. Previous year questions. PYQs 1. CSIR-NET Life Sciences 2019. Which of the following post-translational modifications is primarily involved in the regulation of gene expression by altering chromatin structure? A. Phosphorylation. B. Methylation. C. Acetylation. D. Ubiquitination. Answer: C. Acetylation. Explanation. Acetylation of histones relaxes chromatin structure, leading to the activation of gene expression. 2. CSIR-Net Life Sciences 2018. Which of the following post-translational modifications is responsible for targeting proteins for degradation via the proteasome? A. Phosphorylation. B. Glycosylation. C. Ubiquitination. D. Acetylation. Answer. C. Ubiquitination. Explanation. Ubiquitination involves the attachment of ubiquitin to proteins, marking them for degradation by the proteasome. 3. CSIR-Net Life Sciences 2020. In eukaryotic cells, the attachment of small ubiquitin-like modifier sumo proteins to a target protein is referred to as accumulation. B. Phosphorylation. C. Ubiquitination. D. Acetylation. Answer. Accumulation. Explanation. Sumoylation involves the attachment of sumo proteins to lysine residues of target proteins, influencing their function, localization, and stability. Conclusion. Post-translational modifications are vital processes that control a vast array of cellular functions, from gene expression to protein degradation and cell signaling. Understanding these modifications and their impact on cellular processes is crucial for answering both conceptual and applied questions in the CSIR-Net Life Sciences exam. Mastery of this topic will aid in tackling complex questions related to cellular regulation, protein function, and disease mechanisms. You said. Control of gene expression at transcription and translation level. ChatGPT said. Control of gene expression at transcription and translation level. CSIR-Net JRF Life Sciences. Detailed theory notes with PYQs. Paragraph format. No tables. Gene expression is a process by which information from a gene is used to synthesize functional gene products such as RNA or proteins. In eukaryotic and prokaryotic systems, the control of gene expression is essential for cell differentiation, development, homeostasis, and response to environmental. The regulation can occur at multiple stages, with transcriptional and translational levels being the most crucial control points. By transcriptional level control. The transcriptional level is the primary and most energy-efficient point of gene regulation. This involves mechanisms that determine whether a gene is transcribed into RNA or not, and if so, how much and when. 1. Promoter and enhancer activity. Gene transcription begins at promoters, which are specific DNA sequences where RNA polymerase binds. The strength and accessibility of the promoter determine how efficiently a gene is transcribed. Enhancers are distal regulatory DNA sequences that enhance the transcription rate by facilitating DNA looping to bring transcription factors and co-activators close to the promoter. 2. Transcription factors and regulatory proteins. Transcription factors are proteins that bind to specific DNA sequences near promoters and enhancers. They can be activators, promote transcription, or repressors, inhibit transcription. Their activity is regulated by signal transduction pathways, post-translational modifications, and cofactors. For example, steroid hormone receptors act as transcription factors that bind hormone response elements on DNA upon ligand binding. 3. Epigenetic modifications. Epigenetic changes affect gene expression without altering the DNA sequence. These include DNA methylation, addition of methyl groups to cytosine bases, generally silencing gene expression, and histone modifications like acetylation, methylation. Histone acetylation by histone acetyltransferases HATS relaxes chromatin and promotes transcription, whereas desylation represses transcription. 4. Chromatin remodeling. Chromatin structure plays a critical role in transcript regulation. Chromatin remodeling complexes like SWI, SNF use ATP to reposition or eject nucleosomes, making promoter regions accessible to transcription machinery. 5. Operon model prokaryotes. In prokaryotes, gene clusters called operons, e.g., lac operon, allow coordinated regulation. The lac operon is turned on in the presence of lactose and absence of glucose, demonstrating inducible expression regulated by repressors, inducers, and catalyte activator proteins cap. 2. Translational level control. Even after an mRNA is transcribed, its translation into protein can be finely regulated, especially in eukaryotes. Translational regulation allows cells to rapidly respond to stimuli without the need for new transcription. 1. Initiation regulation. Translation initiation is a rate-limiting step and a major control point. In eukaryotes, ACE eukaryotic initiation factors help in ribosome assembly. Phosphorylation of ACE2 by kinases like GCN2 are perked under stress and activates it, thereby suppressing global translation. This mechanism is crucial in stress responses and developmental control. 2. mRNA stability and degradation. mRNA molecules differ in their half-lives, which affects how protein is made. Regulatory sequences in the 3' untranslated region 3 UTR, such as rich elements, influence degradation by recruiting proteins or mRNAs. Decapping, detonylation, and endonucleolytic cleavage are mechanisms by which mRNAs are degraded. 3. MicroRNAs, mRNAs, and RNA interference RNAi. mRNAs are small non-coding RNAs that base pair with target mRNAs to inhibit translation or promote degradation. In RNAi, double-stranded RNA is processed into siRNAs, which guide the RISC complex to complementary mRNA for cleavage. This mechanism is widely used in gene silencing. 4. Upstream open reading frames URFs. These are short ORFs found in the 5' UTR of certain mRNAs. They can stall ribosomes or alter translation efficiency of the main coding sequence, thus regulating gene expression at the translational level. 5. Riboswitches and translational repressors prokaryotes. In prokaryotes, regulatory molecules riboswitches or proteins can bind directly to the mRNA to block the ribosome binding site RBS, preventing translation. Some proteins act as repressors by binding their own mRNA, a form of feedback inhibition. Biological significance of multi-level control. Energy efficiency. Controlling gene expression at transcription saves energy by avoiding unnecessary mRNA synthesis. Translational control allows rapid response without de novo transcription. Development and differentiation. Sequential gene expression through both levels ensures timely and tissue-specific protein production during development. Stress responses. Cells regulate global protein synthesis during hypoxia, heat shock, or nutrient starvation mainly at the translational level. Disease implications. Dysregulation at either level can lead to cancer, autoimmune disorders, and neurodegeneration. Previous year questions. PYQs. CSIR-Net Life Sciences 2016. Which of the following statements best describes the lac operon regulation? E. coli. A repressed by lactose and activated by glucose. B. Repressed by glucose and activated by lactose. C. Activated by both lactose and glucose. D. Activated only by glucose. Answer. B. Repressed by glucose and activated by lactose. Explanation. Lactose induces transcription by inactivating the repressor. Glucose represses the operon via catalyte repression. Low camp. 2. CSIR-Net Life Sciences 2021. Which one of the following mechanisms contributes to post-transcriptional gene regulation in eukaryotes? A. DNA methylation. B. Histone acetylation. C. RNA splicing. D. Transcription factor binding. Answer. C. RNA splicing. Explanation. RNA splicing regulates gene expression by producing different mRNA isoforms from the same gene. Alternative splicing. 3. CSIR-Net Life Sciences 2017. A2 phosphorylation during cellular stress leads to enhanced global translation. B. Global repression of translation. C. Increased mRNA degradation. D. Enhanced ribosome biogenesis. Answer. B. Global repression of translation. Explanation. Phosphorylated A2 prevents initiation complex formation, inhibiting translation. Conclusion. Control of gene expression at transcription and translation levels is essential for the precise regulation of cellular functions. While transcriptional control determines when and where genes are expressed, translational control fine-tunes how much protein is produced and how quickly the cell can respond to changes. A deep understanding of these mechanisms not only aids in decoding complex life processes but also enables effective preparation for CSIR-NET life sciences questions, particularly those relating to molecular biology, gene regulation, and cell signaling. You said, regulating the expression of phages, ChatGPT said. Regulating the expression of phages. CSIR-NET JRF Life Sciences, ultra-detailed theory notes based on PYQ areas, paragraph format, note tables, maximum content. The regulation of gene expression in bacteriophages, phages, viruses that infect bacteria, is a classic and foundational model for understanding gene regulation, developmental decision-making, and genetic switches. Phages have evolved highly efficient and finely tuned mechanisms to control the timing, sequence, and mode of gene expression during infection of bacterial hosts. These mechanisms are among the most well-characterized systems in molecular biology and are critical topics for CSIR-NET life sciences, particularly from the perspective of prokaryotic gene regulation, molecular switches, and transcriptional control. An overview of phage life cycles. Bacteriophages can exhibit two major types of life cycles. Lytic cycle. The phage hijacks the host machinery, replicates, produces new virions, and lyses the host cell. Lysogenic cycle. The phage integrates its genome into the host DNA as a prophage and replicates passively along with the host until conditions favor a switch to the lytic cycle. Phage lambda lambda is the best studied temperate phage and a key model for understanding regulatory decisions between lysis and lysogeny. 2. Regulatory control in phage lambda. Step 1. Lytic versus lysogenic decision. Immediately upon infection, the phage must choose between two mutually exclusive pathways, lysogeny or lysis. This decision is controlled by a genetic switch involving two major transcriptional regulators. C repressor lambda repressor promotes lysogeny by repressing lytic genes and maintaining its own expression. Proprotein promotes lysis by repressing C expression. 2. Early transcription and promoters. Lambda phage has two early promoters, PL left and PR right, which initiate transcription in opposite directions. Upon infection, host RNA polymerase begins transcription from these promoters. The PL promoter transcribes N gene, while PR transcribes Pro gene. 3. Anti-termination mechanism via N and Q proteins to allow the transcription of delayed early and late genes. Lambda uses an anti-termination mechanism. The N protein produced from early genes enables RNA polymerase to read through terminators, allowing transcription of middle genes like C and C. The Q protein, synthesized later, allows RNA polymerase to bypass terminators and transcribe late lytic genes necessary for virion assembly and cell lysis. This layered mechanism ensures temporal regulation. Early, middle, and late genes are expressed in the correct order. 4. Role of C and C proteins. The C protein is a transcriptional activator that promotes lysogeny by activating pre-promoter for C transcription. High promoter for integrase gene required for integration into the host genome. However, C is unstable and subject to degradation by host proteases. C acts as a stabilizer of C by inhibiting these proteases. The balance between C activity, lysogeny, and pro-lysis is influenced by host cell health. In nutrient-rich conditions, C is degraded, favoring lysis. Under stress, C is stable, favoring lysogeny. 5. Maintenance of lysogeny. If lysogeny is chosen, the C protein lambda repressor is transcribed and binds to operators OR1 and OR2, repressing PR progene and activating its own promoter PRM, forming a positive feedback loop. This maintains the repressed state of lytic genes. 6. Induction of lytic cycle from lysogeny. Under stress, e.g., UV damage, the RECA protein of E. coli becomes activated and cleaves the C repressor. This derepresses lytic genes, allowing transcription from PR and PL and shifting the prophage into the lytic cycle. Regulatory themes in other phages. T7 phage. T7 is a lytic phage that encodes its own phage-specific RNA polymerase. Initially, host RNA polymerase transcribes early genes, including the gene for phage RNA polymerase, which then takes over and transcribes middle and late genes. This demonstrates transcriptional autonomy and progressive control of gene expression. T4 phage. T4 uses modification of host RNA polymerase by phage-encoded proteins to change its promoter specificity during different stages. It also deploys anti-sigma factors to redirect transcription, thus tightly regulating the transition from early to late gene expression. Biological and experimental importance. The lambda switch is a paradigm for binary genetic decisions, used in synthetic biology to construct gene circuits. These systems demonstrate transcriptional logic, feedback loops, anti-termination, and cofactor regulation, making them essential models for gene regulatory networks. Phage regulatory systems serve as models to understand viral latency, host-pathogen interactions, and developmental switches in multicellular organisms. Previous year questions. PYQs 1. CSIR-NET Life Sciences 2014. In lambda phage, high levels of C protein favor a lysis. B lysogeny, C induction, D recombination. Answer: B lysogeny. Explanation: high C levels activate C and integrates genes promoting integration and repression of lytic genes. 2. CSIR-NET Life Sciences 2017. Which of the following statements is true about the lambda phage lysogenic cycle? A crow represses C to maintain lysogeny. B C repressor binds operators and represses its own promoter. C activates transcription from pi and pre-promoters. D C degrades C to promote lysis. Answer: C activates transcription from pi and pre-promoters. Explanation: this is essential for lysogenic entry by producing integrase and C repressor. 3. CSIR-NET Life Sciences 2021. What happens when RECA protein is activated in a lysogenic E. coli infected with lambda phage? A C is synthesized. BC is cleaved. C protein is repressed. D lysogeny is stabilized. Answer: BC is cleaved. Explanation: activated RECA cleaves C, lifting repression of lytic genes and triggering induction. Conclusion: the regulative phage gene expression is a masterclass in genetic control, integrating transcriptional regulation, protein stability, DNA binding dynamics, and feedback systems. From the lambda phage switch to the autonomous transcriptional control in T7, phages offer models to understand developmental phage decisions, genetic switches, and viral latency. These systems form a vital portion of the CSIR-NET Life Sciences syllabus, and mastering their logic is essential to crack high-level questions related to gene regulation, molecular biology, and virology. You said, viruses, ChatGPT said. Viruses, structure, classification, and molecular biology. CSIR-NET Life Sciences, ultra-detailed notes, paragraph-based, note tables, maximum detail, includes PYQs with answers in years. Viruses are acellular, obligate intercellular parasites that can infect all forms of life including bacteria, archaea, and eukaryotes. They are composed of nucleic acid, DNA or RNA, enclosed in a protein coat capsid, and in some cases, an additional lipid envelope derived from the host cell membrane. Viruses lack ribosomes, metabolic machinery, and the capacity for independent induction. They rely entirely on host cell systems for replication, making them important biological models for understanding gene expression, genome replication, and evolutionary mechanisms, especially in the context of CSIR-NET Life Sciences. I structure and genome composition. Viral genomes are highly diverse, ranging from single-stranded or double-stranded DNA or RNA, either linear or circular, and vary greatly in size, from a few thousand bases to over a million base pairs, e.g., in mimiviruses. RNA viruses may be positive sense directly translated or negative sense requiring a complementary strand for translation. Some viruses, like retroviruses, carry RNA genomes but replicate through DNA intermediates using reverse transcriptase. The capsid is made of capsome or protein subunits, assembled in helical, icosahedral, or complex geometries. In enveloped viruses, the envelope contains viral glycoproteins critical for host cell entry, often through receptor-mediated mechanisms. 2. Viral life cycle. The viral replication cycle includes several ordered stages. 1. Attachment adsorption. The virus binds specific receptors on the host cell surface via viral envelope proteins or capsid proteins. This specificity determines the host range and tissue tropism, e.g., HIV binds CD4 plus T cells via GP120. 2. Entry. Entry occurs via membrane fusion, enveloped viruses, or endocytosis, non-enveloped. Some phages inject their genome directly into the host, e.g., T4. 3. Uncoding. The viral genome is released into the cytoplasm or nucleus. 4. Replication and transcription. Genome replication depends on the type of virus. DNA viruses typically replicate in the nucleus using host DNA polymerases, except poxviruses. RNA viruses replicate in the cytoplasm using virus-encoded RNA-dependent RNA polymerases. Retroviruses reverse transcribe RNA into DNA using reverse transcriptase and integrate it into the host genome. 5. Translation. Host ribosomes translate viral mRNAs. RNA viruses with positive sense genomes can directly act as mRNA. 6. Assembly. Capsid proteins self-assemble, and nucleic acids are packaged into virions. 7. Release. Virions are released by cell lysis, non-enveloped viruses, or budding enveloped viruses, during which the envelope is acquired. 3. Viral enzymes and special features. Viruses encode several unique enzymes, such as reverse transcriptase RT in retroviruses, e.g., HIV synthesizes DNA from RNA. Integrase integrates viral DNA into host genome. Protease cleaves viral polyproteins into functional units. RNA-dependent RNA polymerase, RDRP, in RNA viruses for genome replication and mRNA synthesis. Many viruses produce polyproteins from a single long open reading frame, which are then cleaved by viral proteases. IV. Viral classification and families. Baltimore classification. David Baltimore classified viruses based on their genome type and mode of mRNA synthesis into 7 groups. Group 1, dsDNA, e.g., herpes virus. Group 2, ssDNA, e.g., parvovirus. Group 3, dsRNA, e.g., reovirus. Group 4, plus ssRNA, e.g., poliovirus. Group 5, minus ssRNA, e.g., influenza virus. Group 6, plus ssRNA with reverse transcriptase, e.g., HIV. Group 7, dsDNA with reverse transcriptase, e.g., hepatitis B. B retroviruses and provirus formation. Retroviruses, e.g., HIV, carry 2 copies of single-stranded plus RNA and a reverse transcriptase enzyme. After infection, RT converts RNA into complementary DNA, cDNA. Viral DNA integrates into the host genome as a provirus, transcribed along with host genes. Expression of viral proteins and assembly of new virions follows, often latent for long periods. B. Bacteriophages and gene regulation. Bacteriophages, e.g., lambda and T4, are essential in studying genetic switches, transcription regulation, and lytic lysogenic cycles. The lambda phage is particularly known for the regulatory switch controlled by C repressor and CRO proteins, dictating entry into lysogeny or lysis. 7. Viruses and host interaction. Viruses manipulate host cell signaling, shut down host transcription or translation, or mimic host proteins. Many DNA viruses block MHC presentation, helping escape immune recognition. Some viruses encode oncogenes or integrate near proto-oncogenes, leading to transformation, e.g., HPV, EBV, HTLV1. 8. Antiviral strategies and vaccines. Live attenuated vaccines, e.g., MMR. Inactivated vaccines, e.g., polio, influenza. Subunit vaccines, e.g., HBV surface antigen. Nucleic acid vaccines, e.g., mRNA vaccines for SARS-CoV-2. Antiviral drugs target specific viral enzymes like reverse transcriptase, e.g., AZT neuraminidase, e.g., oseltamivir, or protease, e.g., lopinavir. Previous year questions. PYQs 1. CSIR Net Life Sciences 2016. Which of the following viral components is essential for retroviral genome integration into the host DNA? A. RNA-dependent RNA polymerase. B. DNA ligase. C. Integrase D helicase. Answer. C. Integrase. Explanation. Integrase mediates the integration of reverse transcribed viral DNA into the host genome, crucial for retroviral replication. 2. CSIR Net Life Sciences 2019. HIV is a retrovirus. Which of the following statements about its replication cycle is correct? A. Its genome is double-stranded DNA. B. Its RNA genome is translated directly. C. It uses host reverse transcriptase. D. It integrates its genome into the host DNA. Answer. D. It integrates its genome into the host DNA. Explanation. HIV reverse transcribes its RNA into DNA, which is then integrated into the host chromosome. 3. CSIR Net Life Sciences 2022. Positive strand RNA viruses differ from negative strand RNA viruses in that a positive strand RNA viruses carry DNA. B. Positive strand RNA viruses require reverse transcriptase. C. Their RNA can act directly as mRNA. D. They replicate in the nucleus only. Answer. C. Their RNA can act directly as mRNA. Explanation. Positive sense RNA is translated by host ribosomes without needing a complementary intermediate. Conclusion. Viruses provide elegant systems for dissecting fundamental molecular mechanisms of life, from genome replication and gene regulation to protein synthesis and host manipulation. Their minimal genomes encode maximum functionality, making them critical for understanding core concepts in molecular biology, evolution, immunology, and biotechnology. For CSIR Net Life Sciences, mastering viral structure, life cycles, gene expression, and the manipulation of host machinery is crucial to answering high-level conceptual and application-based questions with confidence. You said. Prokaryotic and eukaryotic genes. ChatGPT said. Prokaryotic and eukaryotic genes. Ultra-detailed CSIR Net JRF Life Sciences lecture notes based to advanced level. Maximum word limit. No tables. Paragraph format. Includes 2 CSIR Net PYQs with answers in years. Genes, the fundamental units of heredity, are stretches of DNA that code for RNA or proteins. Although prokaryotic and eukaryotic genes share the core principle of carrying genetic information, they differ significantly in organization, structure, transcriptional regulation, and post-transcriptional processing. Understanding these distinctions is crucial for interpreting gene regulation, expression patterns, and evolutionary adaptations in both domains of life. For the CSIR-NET JRF Life Sciences exam, this topic serves as a high-yield area, especially in the context of molecular biology, genetics, and cell biology. Prokaryotic genes, features and expression. In prokaryotes such as bacteria, e.g., E. coli, the genetic material is typically a single circular chromosome with compactly arranged genes, often forming operons, a key feature distinguishing prokaryotic gene organization. Operons are clusters of genes transcribed as a single polycistronic mRNA, meaning that one promoter and one terminator control the expression of multiple, function-related genes. A classic example is the LAC operon, which contains genes required for lactose metabolism and is regulated by the LAC repressor in the CAP-CAMP complex. There are no introns in most prokaryotic genes, and the absence of a nuclear membrane enables couple transcription and translation. Ribosomes begin translating mRNA while it is still being synthesized. Promoters in prokaryotes include conserved sequences such as the -10 Pritnell box and -35 regions, recognized by the sigma factor associated with RNA polymerase. Transcription termination can occur by row-dependent or row-independent mechanisms. Regulation in prokaryotes is generally straightforward and responds quickly to environmental signals. Regulatory proteins such as repressors or activators modulate transcription by binding to operator sequences, either blocking or enhancing RNA polymerase binding. Eukaryotic genes, structure and expression complexity. In eukaryotes, genes are found on multiple linear chromosomes within a membrane-bound nucleus, and the genome contains vast non-coding regions. Eukaryotic genes are monocistronic, meaning each gene produces one mRNA that translates into a single protein. The typical eukaryotic gene comprises exons coding sequences and introns non-coding sequences. After transcription, introns are removed by the spliceosome, and exons are joined to form mature mRNA, a process absent in prokaryotes. Eukaryotic transcription involves three RNA polymerases: RNA pol I, rRNA, RNA pol II, mRNA, and some snRNA, and RNA pol III TRNA, 5sRRNA. Transcription initiation is highly regulated and requires core promoter elements such as the top box, initiator sequences INR, and downstream promoter elements DPD. Unlike prokaryotes, RNA polymerase II cannot initiate transcription on its own and depends on multiple general transcription factors. GTFs, like TFID, contains TBP, TFIH, and others, forming a pre-initiation complex PIC. Gene regulation in eukaryotes is vastly more complex, involving enhancers, silencers, co-activators, mediator complexes, and chromatin remodeling. Enhancers can act over large distances and may be located upstream, downstream, or within introns. Transcription factors TFs recognize specific DNA motifs and interact with chromatin and the transcriptional machinery to regulate gene expression. Epigenetic mechanisms such as DNA methylation and histone modifications, e.g., acetylation, methylation, further modulate gene accessibility and transcriptional activity. Post-transcriptionally, eukaryotic pre-mRNA undergoes extensive processing, 5' capping, splicing, and 3' polyadenylation, followed by nuclear export. This processing allows for regulatory checkpoints and alternative splicing, which increases proteome diversity. Transcription and translation are compartmentalized, and mRNAs must be exported to the cytoplasm for translation. Comparative overview. While both prokaryotic and eukaryotic genes consist of promoter regions, regulatory sequences, and coding sequences, their genomic context, regulatory complexity, and transcriptional dynamics are markedly different. The presence of operons, lack of introns, and coupled transcription translation are hallmarks of prokaryotic gene expression. In contrast, splicing, chromatin remodeling, and transcriptional activation via enhancer elements distinguish eukaryotic gene regulation. These differences underpin many functional, evolutionary, and biotechnological implications, ranging from genetic engineering in bacteria to gene therapy in humans. CSIR-NET Previous Year Questions PYQs 1. CSIR-NET Life Sciences, Dec 2013. Which one of the following is a correct statement regarding transcription in prokaryotes and eukaryotes? A RNA polymerase in eukaryotes binds directly to the promoter B. Transcription and translation are coupled in eukaryotes. C. Prokaryotic genes are mostly internriched in eukaryotes. Transcription requires general transcription factors for initiation. Answer: D. In eukaryotes, transcription requires general transcription factors for initiation. Explanation: eukaryotic RNA polymerase II cannot initiate transcription on its own and requires GTFs such as TBP, part of TFID, TFIB, TFIE, TFIF, and TFIH. 2. CSIR-NET Life Sciences, June 2017. Which of the following is a correct feature of prokaryotic transcription? A RNA polymerase needs enhancers for initiation. B. Splicing of primary transcripts is essential. CA single mRNA can encode multiple proteins. DNA is processed in the nucleus before translation. And CA single mRNA can encode multiple proteins. Explanation: prokaryotic mRNAs are often polycistronic, meaning multiple genes are transcribed into a single mRNA molecule that encodes multiple proteins. 3. CSIR-NET Life Sciences, June 2021. Which of the following statements best explains the necessity of RNA processing in eukaryotes? A. It enhances ribosomal binding. B. It removes coding regions to prevent clear export. D. It enables maturation and regulation of mRNA. Answer: D. It enables maturation and regulation of mRNA. Explanation: RNA processing, including splicing, capping, and polyadenylation, is crucial for creating a stable, export-ready mRNA molecule that can be efficiently translated. Conclusion: a deep understanding of distinctions between prokaryotic and eukaryotic genes provides critical insight into the regulation of gene expression, the mechanisms of genome evolution, and the molecular basis of cellular complexity. For CSIR-NET JRF Life Sciences aspirants, mastering this topic is essential, as it links seamlessly with high-yield areas like transcription, translation, and gene regulation. With clear conceptual grounding, you can confidently tackle both factual and application-based questions in this domain. You said. Role of chromatin in gene expression and gene silencing. ChatGPT said. Role of chromatin in gene expression and gene silencing. Ultra-detailed CSIR-NET JRF Life Sciences lecture, based to advanced level, no tables, maximum word limit, paragraph format with 2 to 3 previous year questions and answers with year mentioned. Gene expression in eukaryotic cells is a tightly regulated process, and chromatin structure plays a fundamental role in this regulation. Chromatin, the complex of DNA and histone proteins, not only packages the long eukaryotic genome into the confined space of the nucleus but also determines the accessibility of DNA to transcriptional machinery, thereby influencing both gene activation and silencing. Chromatin exists in two major forms: euchromatin, which is loosely packed and transcriptionally active, and heterochromatin, which is densely packed and usually transcriptionally silent. The ability of chromatin to switch between these states underlies the epigenetic regulation of gene expression. At the molecular level, chromatin is organized into repeating units called nucleosomes, each consisting of approximately 147 base pairs of DNA wrapped around the histone octamer, to each of H2A, H2B, H3, and H4. The fifth histone, H1, helps in higher order compaction of chromatin but also represses transcription by making DNA less accessible. Gene expression is largely influenced by post-translational modifications (PTMs) of histone tails, such as acetylation, methylation, phosphorylation, ubiquitination, and sumolation. These modifications are the signals that either relax chromatin structure to permit transcription or tighten it to suppress gene activity. For example, histone acetylation, typically on lysine residues like H3K9AC or H3K27AC, carried out by histone acetyltransferases, has reduces the positive charge on histones and weakens their interaction with negatively charged DNA, leading to a more relaxed chromatin state that favors transcription. In contrast, histone deacetylases (HDACs) remove acetyl groups, promoting chromatin condensation and gene silencing. Histone methylation is another critical modification with context-dependent outcomes. For instance, H3K4Me3 is associated with active promoters, while H3K9Me3 and H3K27Me3 are hallmarks of transcriptional repression. These methyl marks recruit specific chromatin-binding proteins such as HP1, heterochromatin protein 1, and the polycomb group PCG proteins, which help maintain the repressed state. DNA methylation, especially at CPG islands in promoter regions, also plays a key role in long-term gene silencing. DNA methyltransferases (DNMTs) add methyl groups to cytosines, often resulting in the recruitment of methyl CPG binding domain (MBD) proteins, which in turn recruit HDACs and histone methyltransferases to establish a repressive chromatin environment. Chromatin remodeling complexes, such as SWI-SNF, ISWI, ENO80, and CHD, use ATP hydrolysis to reposition or evict nucleosomes, allowing transcription factors and RNA polymerase access to DNA. The SWI-SNF complex, for example, is known for its role in displacing nucleosomes at gene promoters, facilitating transcription initiation. Conversely, polycomb repressive complexes PRC1 and PRC2 mediate gene silencing during development and cell differentiation. PRC2 catalyzes H3K27Me3, while PRC1 binds to this mark in compact chromatin, blocking transcription. The interplay between chromatin modifications and DNA binding proteins defines epigenetic memory, ensuring that gene expression patterns are stably inherited during cell division. This is crucial for maintaining cell identity and for processes such as X chromosome inactivation, genomic imprinting, and stem cell differentiation. Disruptions in chromatin regulation often lead to diseases such as cancer, where aberrant silencing of tumor suppressor genes or activation of oncogenes occurs. In summary, chromatin serves not just as a passive structural scaffold but as an active and dynamic regulator of gene expression. Its modifications and remodeling determine whether a gene remains accessible and active, or condensed and silenced, highlighting its central role in transcriptional regulation, developmental gene control, and epigenetic inheritance. CSIR Net Life Sciences Previous Year Questions PYQs 1. CSIR Net Life Sciences, June 2016. Which of the following histone modifications is most commonly associated with transcriptional activation? AH3K9Me3. BH3K27Me3 CH3K4Me3 DH3K9Me2. Answer: CH3K4Me3. Explanation: triangulation of lysine 4 on histone H3K4Me3 is a well-established marker of transcriptionally active promoters. 2. CSIR Net Life Sciences, DEC 2017 DNA methylation in promoter regions generally leads to activation of transcription B increased histone acetylation C gene silencing. D increased transcription factor binding. Answer: C gene silencing. Explanation: DNA methylation at promoter CPG islands leads to recruitment of repressors like MBD proteins and HDACs, resulting in chromatin condensation and gene silencing. 3. CSIR Net Life Sciences, June 2018. Which one of the following protein complexes is involved in chromatin remodeling to facilitate transcription activation? APRC2. BH circa SWI-SNF DDNMT. Answer: CSWI-SNF. Explanation: the SWI-SNF complex is an ATP-dependent chromatin remodeler that shifts or removes nucleosomes to allow transcription machinery to access DNA. Conclusion: chromatin plays a master regulatory role in the life of a gene, from its activation to its silencing. Its structural forms, post-translational histone modifications, DNA methylation status, and interaction with remodeling complexes determine whether transcription can proceed. This layered regulation ensures that genes are expressed in the right cell, at the right time, and in the right amount. For CSIR Net JRF aspirants, mastering chromatin biology is essential, as it links directly with key themes in epigenetics, molecular biology, development, and disease, all frequent areas for conceptual and application-based questions in the exam. Search. Breeze.